So I want to thank the organizers for giving me the chance to talk to you today about what we can learn about aging from the roundworm C. elegans. So one of the oldest questions humans have grappled with is the question of why we age. And even though the causes of aging are still largely mysterious, the effects of aging are well known, such as the loss of energy and motivation that comes with getting old. And a related question is why some people live long and age in a healthy manner, whereas others seem to age more quickly and suffer from debilitating illness. And these questions are a pressing concern, not only from a personal standpoint, but from a societal standpoint, because the median lifespan in developed countries has been steadily increasing since 1840. And so the fact of our aging population has significant implications for the demands that will be put on our healthcare system in the coming years, especially in light of the fact that aging is the #1 risk factor for diseases like heart failure, neurodegeneration, and cancer. So for these reasons, it's crucial that we get a better understanding of the causes of functional decline so that we can reduce the suffering inflicted by aging and diseases associated with it. Now, one clue about why individuals age differently is that similarly sized animals like mice and bats can have drastically different lifespans. And one of the primary differences between these animals is that they have different genes. This suggests that genes might play an important role in regulating the rate of aging. And if we could identify those genes and how they work, we could understand the keys to longevity and healthy aging. So the idea that genes are critical in regulating aging was proven right 30 years ago when it was discovered that mutations in a single gene could double the lifespan of the roundworm C. elegans. So to illustrate this, on day 1 of adulthood, N2, which is the wild-type strain, and DAF2, which is a mutant, both crawl with elegant sinusoidal body bends. But a few weeks later, the N2 worm is near death, whereas the DAF2 worm can still crawl around normally. So it seems like the DAF2 mutant is aging more slowly. And this work showed that a single gene could dramatically alter lifespan. And since this discovery, many more genes have been discovered that influence lifespan. So for example, pathways such as dietary restriction, insulin signaling, sirtuins, and mTOR have all been shown to modulate aging in species across the evolutionary tree, including in primates. And some of these pathways have even been proposed as potential therapeutic targets to reduce aging morbidity. So for example, drugs like metformin and rapamycin have both been shown to slow aging and reduce the incidence of age-associated illness in worms and mice by affecting the function of these conserved molecular pathways. Now, the organism in which many of these pathways were first discovered and best understood is the roundworm C. elegans, which is the model organism our lab studies. And over the past few decades, C. elegans has emerged as an important model to study the biology of aging, especially because of its short lifespan and the similarity of genetic pathways between worms and mammals. Now, the traditional way lifespan is measured in worms is by manually counting the number of animals alive in a population over time. But these manual lifespan assays have a number of limitations. They're low throughput, time-consuming, and only measure time of death for each worm, which means they lose all information about each individual's life history and therefore the aging process itself. So questions like how lifespan relates to other aspects of aging, such as the fraction of life spent in a state of good health, are often ignored because of the difficulty of manually measuring these long-term phenotypes. So we thought that the limitations posed by manual lifespan assays could be addressed by automating these measurements. So we set out to design a device capable of long-term monitoring of worm health over their entire lives so that we could learn more about the aging process beyond what these standard lifespan assays could teach us. And to do that, we began by considering what features an ideal method would have. So first, we wanted a way to uniquely monitor many worms over long time periods without animals mixing together, so that we could record lifelong aging measurements per animal. The method also had to be able to sustain worms for their entire lives, which can range up to 2 months. And then lastly, we wanted the method to be scalable so that we could use it to monitor small numbers of worms, up to hundreds or thousands, to enable screens. So after many iterations, we eventually came up with something that fulfilled all of our design criteria. And the method we came up with, we call the worm motel. Now, the worm motel consists of an array of 240 individual wells, each of which houses a single worm, which allows us to uniquely monitor each animal's behavioral health over its entire life. And we designed the worm hotel to be compatible with standard lab automation tools, which enables us to scale up and perform genetic and compound screens for modulators of aging. So to prepare an experiment, each well of the device is filled with standard agar and bacterial food, followed by a single worm. And then we can image the device under a camera for up to 2 months. So this is what it looks like on day 1 of adulthood, with each of the 240 wells filled with a single worm. And the way it works is we image the device twice a day for 30 minutes, and at the 15-minute mark, a bright light stimulus turns on for 10 seconds, which serves to wake worms up and allows us to measure their maximum capacity to move, which we then take as a proxy for how healthy each worm is. So here's a closer look at 9 worms on day 1 of adulthood. We can better see the increase in movement after the blue light. So these worms are all looking very healthy. However, 2 weeks later, notice how much less healthy worms look than they did as young adults. They barely move at baseline and barely respond to the blue light. So these worms are ready for assisted living. Okay, then to process the images, we can use a simple machine vision algorithm to quantify how much each worm moves over time. What we do is subtract subsequent images to generate a difference image, and then count up all the pixels where a change in pixel intensity occurred, and call the resulting number the worm's activity. So the more a worm moves between 2 frames, the more pixels will have changed, which corresponds to a high activity value, and the less a worm moves between 2 frames, the fewer pixels will have changed, which corresponds to a lower activity value. And then we can just plot activity over time for each worm. So here's an example of one worm's behavior trace over its entire life, where you can see we can automatically calculate time of death as the animal's final stimulated movement. And then to summarize data for many worms at a time, we can plot behavior in a heat map, where hotter colors indicate more movement, and each row shows the behavior of one animal over its entire lifespan. So to recap, what I've shown you so far is a device called the Worm Hotel our lab developed to automate aging measurements in C. elegans, which allows us to move from experiments that record only time of death to experiments that record behavioral health in addition to lifespan automatically for many animals at a time. So now I just want to give you a sample of some of the questions that our lab's addressing about aging using the Worm Hotel. So for example, in a genetically identical population, some individuals live a short amount of time, whereas others live a long amount of time. So even in a population composed of genetic clones, there's still a large degree of variability in lifespan. So we wondered what difference, if any, there was in the aging process between individuals that live a long amount of time versus individuals that live a short amount of time. Before we look at the data, we can conceptualize what the differences might be in 3 ways. First, short-lived individuals might begin their lives at a lower level of health compared with long-lived individuals, and then decline at the same rate, so they die earlier. Or both groups might begin life at the same level of health, but short-lived individuals decline at a faster rate, causing them to die earlier. Or third, both groups might begin life at the same level of health and decline at the same rate, but short-lived individuals die prematurely for some reason. So I compared the behavioral decline of the shortest and longest lived individuals in a wild-type population. And what I saw is that both groups begin life at the same level of activity or health, but short-lived individuals decline at a faster rate and die prematurely relative to their long-lived siblings. So our data is consistent with a combination of these last 2 models. I then wanted to see if the same phenomenon could be observed in other genetic backgrounds. So I looked at the short-lived strain DAF16 and the long-lived strain DAF2 and broke up behavior based on lifespan. Once again, I saw that short-lived individuals declined at a faster rate and died prematurely relative to their long-lived siblings. So since we know the differences between short and long-lived individuals are not caused by genes, we're now trying to use RNA sequencing to identify what the potential gene expression differences might be that cause the differences in functional decline and lifespan. Okay, so finally, just as previous lifespan studies in worms have improved our understanding of human aging. we'd like our work describing aging to eventually lead to results that are translatable to the clinic. So with that in mind, we'd like to understand the links between lifespan and health span, which is the amount of time spent in a healthy state. So we expect some genetic mutations to lead to moderate health span, maybe 1/2 to 3/4 of life is spent in a healthy state. Other genetic conditions might lead to a low health span, where only a low fraction of life is spent in a healthy state. There might be some conditions, though, that enable animals to have very high health spans, where almost the entire life is spent in a healthy state. And these hypothesized conditions would be especially attractive as potential therapeutic targets. So to begin addressing this question, I used the worm motel to calculate health span for a number of strains with differences in lifespan relative to wild type, shown in black. And I found that some strains increased lifespan without increasing health span. Other strains that increased lifespan while actually decreasing health span. And sure enough, some strains that were able to increase both lifespan and health span. Ideally, though, health span would equal lifespan, so that an animal's entire life could be spent free of age-associated morbidity. So, to that end, our lab developed a robotic plate handling system that can automatically monitor hundreds of worm motels at a time, which we're now using to search for genes that increase the fraction of life spent in a healthy state. So, as I said earlier, most studies of aging to date have focused only on lifespan. But there's still a lot we have to learn about the aging process itself, and we need to continue to refine our studies of aging to understand the links between increased longevity and quality of life. And hopefully I've convinced you that the worm motel is a useful tool that can improve our understanding of the aging process. So with that, I just want to thank the organizers one more time for giving me the chance to talk, my advisor Chris, my thesis committee, our funding sources, and you for your attention.
Any questions for Matthew?
That was really cool system results. I was really struck by your kind of layering of all the lifespan of the individual worms on the one plot, and that, at least based on the heat map you had, almost all of their behaviors seemed to drop at similar amounts of time. So the longest lived individuals were living at much lower rates, and particularly that was true in either DAF2 or DAF6, right. That you have this long extension of span, but they're near the bottom in terms of their utility.
Right.
So you only showed us one example where you got some increase in health span as well, but have you found any ones that have that longer health span, and can you isolate those strains and propagate that at all? Or?
Yeah, so we've just started the screen. We haven't found anything besides TAX4 was the one example that was able to increase both health span and lifespan. We do know some genetic pathways, particularly EGF signaling for whatever reason, is able to increase health span without really affecting lifespan that much. But yeah, it's kind of interesting that all these long-lived individuals, even within a wild-type background, the longer lived the individual is, they seem to not really increase their health span so much as kind of just prolonging the kind of senescent phase. And we're not really sure why that is, but that's something we're really interested in figuring out.
So I'm going to, ask a totally unreasonable question just to let you know.
Okay.
So I look at the genetics of the genes, and that looks like a black box to me. If you told me that this protein wasn't being produced or some other thing wasn't happening, I might have no idea. I would have no idea what that really meant for the life of a worm. So I'm going to make, this is not meant as a joke, but if you were to autopsy a worm, could you figure out the cause of death? Okay, what causes death in worms?
So this is kind of an open research question because people don't necessarily know for sure, but what has been studied before is people looking at worms that seem to be near death. And what people think is that it's kind of spontaneous membrane rupture of the cell membrane. So there's 1,000 cells in worms, and basically over the aging process, the integrity of those membranes deteriorates, and that seems to be the cause of death. When the membranes rupture irreparably, the animal kind of dies quickly.
So all worms die of the same cause? It's just eventually when?
Well, I think the cell membranes are kind of the limiting factor on whether an animal is alive or not. So when the membranes rupture, when the skin ruptures, basically, nothing else can continue. So it's kind of the limiting
So they
factor
get
of
to
what's
that
keeping
state
it
of membrane rupture through different paths?
Say again.
Do they get to the state of membrane rupture, global membrane rupture through different metabolic or other paths?
So that hasn't been studied. So it might be that some kind of genetic conditions cause the membranes to maintain their integrity longer relative to other tissues. But that hasn't been studied. It was only studied in wild type. So it's a good question, though. It's a really good question.
Any other questions for Matthew? All right, let's speaker thinker one more time. Okay.